All right, let's talk about the application of using an epoxide. All right, this is one of the many applications. This one gonna be a little bit on the medical field side. Right? If you were becoming a doctor, medical tech, or medical assistant, so you're probably familiar with this. But I mean, in any field, we use quite a lot of this one to kill bacteria. Actually, all right. So ethylene oxide look like this. All right, so high ring strain, high torsional strain, really reactive. We use this guy in a solution to kill bacteria, microorganism. Right? In surgical instrument, for example, this guy use that in surgical purpose and put into the container and then add the solution of the ethylene oxide. Right? Now, how does this work? Let's take a look at the dirty, let's say surgical knife. Okay, let's say after use, bacteria are like floating around, so we need to be clean before the next use. So we make an ethylene oxide solution, dump this guy in there. All right, so the bacteria are gonna say, uh-oh, kind of cold, nothing happened. Well, here's the thing though, this is in a solution, everyone floating a solution. Or inside the bacteria doesn't have ethylene oxide in it. What's gonna happen? Well, kind of osmosis, kind of similar to that. So it would diffuse to inside a cell. Right, inside the cell of this bacteria. All right, let's take a look at inside the cell. What's inside the cell? Even in the nucleus, so we have a DNA. So we just a lot of stuff, but we target DNA here. All right. So you guys familiar with the DNA structure? So double helix from the interaction with two strand from interaction with the hydrogen bonding A, T, C, and G. Look something like that. Okay. Now, from research, always all the nitrogen and oxygen could act as a nucleophile, but the most nucleophilic nitrogen is this nitrogen, the nitrogen number 7 of guanine. Okay, and this guy can go, of course, will be alkylated by the epoxide, so the epoxide gonna go attached onto that nitrogen. Now, the question is, what does this alkylated guanine do? Does it kill bacteria? Like how? So it's just so small. Well, here's what it works. So let's talk about the a normal function of a DNA. And you guys have heard about the transcription, translation, replication before, right? When all this process happens, the first event that need to happen is to separate the two strands of the DNA. Okay? And the enzyme who does that called helicase. So we zip up, so basically separate two strands. Like zip up right here, and that's allowed a bunch of enzymes that help replicate or copy each strand of the DNA. Okay, helicase is gonna keep open, right? Separate the two strands. The other enzyme and other stuff gonna help replicate. Eventually, you're gonna get two identical of DNA strands. All right, and this for this process specifically we call DNA replication, which is crucial for the cell survival. Now, what does epoxide do? Right now we add an epoxide, so epoxide phase with the DNA. The guanine gonna get alkylated, so it get attached on guanine. Some of them, not all, but yeah, some of them get attached. Okay, so what happened is, if you still remember that the enzyme and the substrate, so right now the enzyme is at the substrate, have to be they have to fit, they have to be compatible, right? They have to be, was it like an analogy of glove and hand? They have to fit like that. But now when guanine have the things coming out, which is the epoxide, right? This helicase cannot or does not recognize this guy as a substrate because this is a totally different compound now. Well, what happened? Helicase cannot work. When helicase cannot work, DNA cannot be separated, no DNA replication, and cell die basically, right? The body or the cell will realize that, well, this cell is useless, so it will go to programmed cell death. We call it apoptosis. All right, so that's how the epoxide kills the bacteria. All right, let's go further to sulfides and thiol.